Good morning, everybody, and welcome back to BioSC 140 Human Physiology. This video is going to be the experimental procedures video for Lab 27, renal function. So in this lab, we're doing lots of different activities. There's a large number of shorter experiments that we're doing within Lab 27. Remember, for the quiz, I expect you to be familiar with the experimental procedures for all the labs. So we're doing FOX 9.1, FOX 9.2, calculations and concepts will be covered on the quiz, and FOX 9.3, clinical examination of urine. You're in for a great lab today. All right, so when working with urine, we use universal precautions. We assume all material that we're working with today to be hazardous. Only handle your own urine. Please wear gloves and properly dispose and clean of any contaminated materials. Urine may be rinsed down the sink with large amounts of water. That's in the bathroom. You can actually do it in the dump it into the restroom, into the toilet, and then you can rinse it out within the sink. Soft contaminated materials can go in biohazard bags, so the soft container for biohazard. Reusable contaminated equipment should be washed with Amphil. Always wash your own contaminated materials. No eating or drinking even water in this lab. If you have a water bottle, please keep it out in the hallway. Wash your hands, go outside, and drink out there if you need to. All right, so let's get into it. So FOX 9.1, renal regulation of fluid and electrolyte balance. So our kidneys do a lot of different things, and a couple of things they do is they regulate fluid levels. When we get a little bit more into our cardiovascular system, we'll see that the long-term, the way that we maintain blood pressure in like a long-term manner is by maintaining proper fluid levels within our body. And so maintaining the right level of fluid, our kidneys have a very large role in that, in maintaining blood pressure. And also electrolyte balance. So what do we do for this part? Well, everybody goes to the restroom and pees in a cup. The more the better. And on the cup that we pee into, there are little hash marks that are volume hash marks. That's how we find out the volume of urine within the cup. Now, this is a clinically important value, and we will be doing some calculations with this number later on in the lab. So the volume, we get the volume just by the hash marks on the container. We're then going to calculate pH of our urine, and to do that, we're going to use a pH strip. So it's a little piece of paper with a colored part on one end. It's a thin strip of paper, and we're going to dip that into our urine and pull it out. And when we pull it out, we're going to compare the color that the tip of the piece of paper is with a reference color. And depending on the color it changes, it'll signify one pH or another pH. We'll know the level. For specific gravity, so specific gravity is density relative to water's density. So it's the density versus the density of water. Water has a specific gravity of 1.0. If something's more dense than water, it'll have a gravity of more than 1.0. And urine is water with stuff dissolved in it that makes it a little bit more dense. So normal is 1.003 to 1.04. And the way that we're going to figure out specific gravity is by using something called a refractometer. And that's this device in the middle up here. This device in the middle up here. So the way it works is you put a drop of urine on this blue part right here where the pointer is. You then close the lid slowly. You don't want anything to spill, so close it slowly. You then look into this side of the refractometer and you point this side at a light, so like outside the window usually. And it's kind of like using a telescope. So you look in this side and you look over here, and you're going to see a scale like this. I mean, this is like a temperature scale, but it looks like, you know, a line with some hash marks and the numbers next to those hash marks. And you'll actually see a line where the specific gravity is. So look at the light, look to the left and record the number. You'll see a line where that's next to a number. Please clean and put those into the biohazard. For chloride concentration, it's important that we dispose of silver waste properly. So silver waste, there's actually going to be a special jug in the fume hood in the back of the room where we're going to dispose of this waste. So we're going to take a test tube and we're going to put 10 drops of urine in it and then 1 drop of potassium chromate. We're then going to add 1 drop of silver nitrate, Ag is silver nitrate, and swirl. We're going to count the number of drops it takes to become rust colored, and then we're going to multiply by 68. So we're going to add 1 drop of silver nitrate, shake. 1 drop of silver nitrate, shake. 1 drop of silver nitrate, shake, until it becomes a rust color like in this photo in the top right. And so we're going to take the number of drops that were required to become rust colored and multiplied by 68. So volume depends on expected values. So volume, it depends on how long it's been since you used the restroom, how much water you've drinking. I feel like students have a pretty good idea of what can affect how much urine you produce, you know, how much water you're drinking, how long has it been since you went to the restroom. Typically 20 to 200 milliliters is a typical range. pH, 6 is average, but normal could be anywhere from 5 to 8. So there's a wide range of normal pH. Specific gravity, it's more than 1. Remember, 1 is pure water and urine is water with stuff dissolved in it. So 1.003 would be dilute urine. So if you've been drinking a lot of water, not really eating too much. And if you haven't been drinking a lot of water, 1.04 is kind of the high end of the typical range. Chloride has a wide range also. So typically it's between 1 and 40 drops. And, you know, so it could be anywhere from 68 to 2,720 milligrams per deciliter, a wide range for chloride. Next up, we're going to do a little bit more clinical examination of urine. So we're going to use chem strips to look for protein, glucose, ketones, blood, and bilirubin within our urine. So the way this works is we don't spray urine in lab. We actually just take the strip and we dip it into our sample. So the strips look like this. They look a lot like a pH strip. And so we'll put a little bit of urine on the sample, on the strip, and we'll see what color it changes. So there's a different spot for each one of these values. You know, there's a different spot for protein, for glucose, for ketones. You can see the different colors on the strip. And then we'll wait 1 minute, and then we'll compare the colors that are on this strip with our reference right here. And you can see how the different colors that it changes will correspond with different concentrations of each one of these values. So one color will correspond with one concentration of protein, another color will correspond with a different concentration of protein. And so we'll do that same process for all 5. Next up, we're going to look at urine sediment. So typically we do this one per group, so 1 sample per group. Up to this point, everybody does all parts. We're going to fill a plastic tube halfway full with urine. We're then going to centrifuge it for 5 minutes, make sure the centrifuge is balanced. We're going to decant all of the liquid off out of the, or pour all of the liquid out of the container. And you'll see a little tiny pellet of solid stuff at the bottom of the container. We're going to add SETI stain, which is going to stain those things on the bottom of the tube so that we can see them. And then we're going to add them to a slide and look at them underneath a microscope. Now, this is actually really cool. And we're going to compare what we see with pages 354 to 356 in our lab manual. And we're going to see what solid sediment is in our urine. All right. If you have any questions, as always, please reach out to me. I'm here to help, and I will see you in the next video.